Hey everybody, my name is Jason Walker. I'm a science coordinator with the National Math and Science Initiative, and this screencast is the second in a series on botany to help us get ready for the AP Biology exam, and we're going to center this one around growth and development of plants. So, plants are able to grow throughout their lifetime due to the presence of undifferentiated tissues called meristems. Now, when we look at this word, undifferentiated, we are reminded of the own stem cells within our bodies. They're not nerve cells, they're not muscle cells, they're not skin cells, that type of thing. They're undifferentiated. Okay? And if we remember from the last screencast, we talked about that most cells within a plant are totipotent, which means that they have the ability to take on the characteristics of other differentiated cells within that plant. So if it needs to be a root, it can be a root. If it needs to be a stem, it can be a stem, that type of thing. Okay? The two meristems that we are going to concern ourselves with are apical and lateral meristems. Apical meristems are going to give the plant primary growth. And they're going to cause the stem of the root to grow longer. They're going to give it length, okay? And they are located at the tips. And lateral meristems are going to add secondary growth. And they're going to cause that plant to increase in girth or width, okay? So if I have a tree coming out of the ground, okay? Now, here's what I'm going to do. I'm going to put a nail right there. And then I'm going to wait 25 years. In 25 years, I'm going to get all kinds of growth, hopefully. Okay? And this thing is going to grow. It's going to flourish. Now, we ask ourselves, where is that nail after 25 years? Apical meristems. Where are those apical meristems? They're at the tips. Right? So, where's the nail? It's right where we left it. Right there. It's not up here somewhere. Right? Because the growth isn't occurring from down here. Awesome. All right. Well, now let's talk about lateral meristems, because if this is like an oak tree or something like that, and we get that secondary growth, that nail might be inside that bark. It might be inside in that secondary xylem. Okay? Have you ever seen those things? Basically, they sell them to put on your tree, like nail them to your tree, and they're like little eyes and a mouth. And after a few years, that secondary growth will actually grow over those eyes, like around that mouth, and it actually looks like, hey, there's a face on my tree. So let's talk about reproduction in angiosperms specifically, right? So this is the anatomy of a flower. Look, it's prior knowledge, but we're going to do a quick brief review here. This idealized flower has male structures called the stamen, which is composed of both an anther and a filament. The anther is the site of pollen production. So what do you think this filament is doing? I mean, look at the evolutionary advantage that filament gives. Okay, you've got a pollinator that's coming along and landing on this flower, right? Well, if that anther is way down there by the receptacle, okay, down here nestled next to the ovary, the chance of that bee or hummingbird or whatever getting into that pollen on it to then go and pollinate another flower is kind of low. But look what that filament does. It just kind of presents that anther out, right? Well, let's look at the female part of the plant, called the carpels, sometimes called the pistil, Pistil, okay? You have the stigma, the style, and then you have the ovary, okay? So, do you remember alternation of generations? This is what it looks like inside of an angiosperm, okay? This is the life cycle of an angiosperm. It's a great overview of the reproductive process. So, again, just to remind you, you do not have to memorize any particular life cycle i.e. alternation generations of any of the 4 different types of plants, but just to understand the basic concept of this alternation between haploid and diploid. Okay, and what we see here is that fertilization is going to occur when the male gamete fertilizes with the female gamete to form the plant embryo. Now, at that point, this zygote is now diploid, right? Our sperm and egg were haploid. Okay. Now, the next 3 slides are going to cover a concept called double fertilization. This double fertilization is unique to angiosperms. Okay, though the details of particular reproductive cycles of plants and animals are no longer specifically covered on the AP Biology exam, double fertilization, it's so crucial to the evolutionary success of angiosperms that we must visit it and discuss it for the exam. So here we have the carpel or the pistil. Okay, you have the stigma, style, and the ovary, and we can see polar nuclei in here, and we can see the egg. Okay, so if the pollen tube, if this pollen grain basically germinates, it's going to grow this. It's going to start growing mitotically down. It's going to grow this pollen tube, and I want you to pay special attention to these 2 sperm that are coming down with that. Okay. The pollen tube is going to discharge the 2 sperm into this female gametophyte called the embryo sac within this ovule. Okay? And basically, one sperm is going to fertilize the egg, which forms the zygote. The other sperm combines with the 2 polar nuclei of the embryo sac's large central cell, forming a triploid cell. This is going to develop into nutritive tissue called endosperm. Now, these 2 events comprise what we call double fertilization. You fertilize the zygote, and you've combined that other sperm with the 2 polar nuclei, making endosperm. Okay, this is double fertilization, and it is very unique to angiosperms. Now, we think of endosperm. It is a very important component of our human diet worldwide. Okay, for instance, let's take wheat. For an example, the endosperm is out of wheat. The endosperm is selectively taken out in the processing of this food. We call it white flour. Okay, the embryo itself, which we call germ, the seed coat, which we call bran, are removed. Okay, now if you keep the embryo and the seed coat, the germ and the bran, you keep those in there. Now we call that whole wheat, right? Some other examples of endosperm would be the milk or the meat, quote unquote, in a coconut. Also, the nutritive tissue inside of corn kernels. That's endosperm as well. Let's discuss the coevolution of these pollinators and flowers. Okay? So, these pollinators are flower specific. Okay? These pollinators will only pollinate certain types of plants, which have certain types of flowers. The flowers are going to evolve along with these pollinators. Just for some examples, butterflies see yellow, thus the flowers they pollinate are yellow. Moths are out most frequently at night, so therefore they pollinate white flowers that are fragrant. Bees see yellow, blue, and UV light. The flowers they pollinate are yellow or blue, and you can see that UV radiation. Hummingbirds need a lot of sugar, and they see red. They pollinate flowers that are red and usually have these significant nectar reserves. Okay? Bats and possums are nocturnal and pollinate flowers that are fragrant and attract insects. Okay? The pollinators and flowers have co-evolved together. Let's take a specific example of this relationship. For example, the California buckeye tree is pollinated only by native California honeybees that are immune to the neurotoxin found in the pollen. Other honeybees are sensitive to this pollen. Okay, so only these California honeybees are immune to this neurotoxin, so they're able to actually pollinate that tree, the flowers on that tree. Some orchids actually resemble insects, so male insects will come along and they'll actually try to mate with these orchids. In effect, they are actually involved in the pollination of these flowers, right? So here let's look at the structure of a mature seed. Okay, you see a seed coat, you see other structures within that, basically associated with endosperm, the different parts of the embryo, your first seed leaf, your first root, that type of thing. Okay, a seed consists of a dormant plant embryo. Okay, it's surrounded by its food supply, the cotyledons, the endosperm, or both. The seed is surrounded by this hard protective seed coat. Okay? Dormancy describes a period when the embryo within a seed stops growing and its metabolism nearly ceases. This is an adaptation for tough times, whether that be drought or freezing winters, that type of thing. Certain environmental conditions can trigger the seed to break this dormancy. Now, it's important. The timing of seed germination and its reliance on both temperature and water are specifically tested on the AP Biology exam. There's two types of germination. Okay? Germination begins with imbibition, the uptake of water by a dry seed. Rupturing of the seed coat triggers metabolic changes that allow the plant embryo to resume growth. Now, the timing of seed germination and its reliance on temperature and water, again, just to point out, these are specifically tested on the AP Biology exam. Okay? Low water potential of the seed triggers this imbibition. Okay? In A here, we see a common garden beans. We see some common garden beans. Okay? The straightening of a hook on the hypocotyl pulls the cotyledon from the soil. Okay? In maize and other grasses that we see down here in B, the shoot grows straight up through the tube of the coleoptile. Okay? Breaking dormancy is a phenomenon attributed to enzymes and hormones within these seeds. The essential seed-related anatomy terms are cotyledon, endosperm, and radicle. So, do dry seeds respire? Yes. Well, unless they've been suffocated in a plastic bag or become completely dehydrated. Finally, let's just look at some adaptations for seed dispersal. Okay, how do these seeds get out into, how are they propagated out away from the mother plant, right? And again, you wouldn't need to memorize all of these, but be able to discuss one or a few. Right? So first we see over here some are wind, like a dispersed, like milkweed or a dandelion or maple. You've probably seen these little helicopter guys coming down to the ground, right? Maybe they're propagated by animals. We have beggar ticks and sandburrs and a blackberry. These things have some spines on there, and they're going to be able to just stick to animals that are walking by, right, that are going to basically go disperse that somewhere else. Even by water, you see a lotus, cattail, even a coconut. What most people think is the coconut is actually the seed, right? And so that's within this big hairy fruit. And that's going to be able to just bob up and down in the waves until it lands on some ground and then be able to grow from there. Some flowers just burst open, okay, like a violet or witch hazel or jewelweed, right? They just burst open and the seeds go flying, right? Some of them, we put these seeds in these fruits that humans like beans and wheat, all right, and cherries. These things have seeds unless we've genetically taken them out, obviously, but we can disperse that. We can do that. Again, please make sure that you go through this this screencast and all screencasts as many times as you need to. Stop, take some notes, ask some questions, ask your teacher, and I really do believe that this is going to help you with the growth and development of specifically the angiosperms on the AP Biology exam.